Hello everyone, my name is Camille Champion. I'm in a postdoctoral position under the supervision of Emmanuel Le Chatelier, Mandra Maria Dassou, and Magali Berlin in Metagenopolis, a unit of INRAE. And today I will talk about one network to rule them all. In recent years, research has been significantly directed toward the role of intestinal microbiota on health. Healthy individuals exhibit well-balanced microbial community, whereas diseases are associated with dysbiosis and disruptions of the communities. The microbiota is typically structured with local subcommunities called microbial guilds that you can see here, composed of bacterial species interacting with each other and sharing a common role in the host ecosystem. Their identification is thus crucial to make a link between taxonomy, ecosystem, health, and to identify keystone species. Formally, microbial interactions can be modeled by networks comprising nodes and edges representing the n observations of the p quantitative variables composing the system. The main issues with inferring a microbial network is the high dimensionality with p larger than n, the sparsity, because we want to build a network with few edges, and the complexity of microbial abundance data with big heterogeneity, heterodicity, and extreme variability. The main objective of this work is to build a consensus network from microbiome data that yield testable hypotheses. Classical networks inference methods applied on microbial abundance data can lead to poorly reproducible and sparse graphs. We thus fix as reference a network inference pipeline adapted to microbial abundance data. So you take an abundance table and then it transforms them in three ways, using measures to take them to the Gaussian, using distributions and models, using models based on latent variables. It then applies network inference approaches based on Gaussian graphical models to estimate the precision matrix representing only the direct interactions of the system. There are two different approaches, first penalized approaches and pre-averaging approaches. Then it fixes a stability threshold to select the edges composing the final graph. We selected 8 different inference network methods that are just here, based on distinct mathematical hypotheses which fall within this pipeline. Applied individually, all generate very different networks, leading to a lack of robustness and reproducibility. We hence decided to select the edges representing a high selection frequency in all individual methods, and then to calculate a consensus frequency by aggregating then these frequencies using min, norm2, IVW, and min3 to get the final network. The proposed method is then applied to simulated data from the empirical dataset cited in Queens, and all articles using the methodology are described in June 2020, which is based on reshuffling an original dataset to preserve the peculiarities of count data. The empirical dataset studied in Queen correspond to stool samples from 216 Chinese individuals sequenced using the whole metagenome sequencing technique. Among the population, 102 individuals are healthy and 114 suffer from liver cirrhosis. The final abundance table is then of size 1990 MSP by 216 individuals, records all the normalized abundance. To assess which tool inferred the most accurate networks and to explore how sample number or input settings affect tool performance, We restricted the study to the disease samples and the species with a prevalence higher than 50%. The first experiment here is conducted with n is equal to 100 to prove the quality of both individual and aggregated frequency. Here you have the different aggregated methods and the different individual methods in terms of precision and recall. Comparing aggregated frequency to individual ones, we see that the aggregation with min norm 2 and min 3 that are here present among the best precision levels and among the worst recall values. This illustrates how they lead to sparser networks and with a high precision. The second experiment here, conducted on liver-cirrhosis dataset, aims to construct a consensus network to identify robust and reproducible edges implied in liver-cirrhosis development. Using the mean aggregation network inference and clustering algorithm, one guild drew our attention as it contains all species known in the literature to be associated to liver cirrhosis. So you have the list of the species just here. Hence, the one net consensus network inferred more true edges than any method taken individually, keeping the best edges of each method and uniting the distinct mathematical hypotheses. This study, which is available on bioRxiv, also opens a new insight in microbial guilds discovery related to a disease using robust network inference and clustering methodology. If you are interested in this work, let's take a look at my poster in the session NetBio of ESMB ECCB conference. Thank you for your attention.